Welcome to the first video on the media for Lab Unit 3, and this video is going to talk about MacConkey Agar. We normally just refer to these as MAC or MAC plates because MAC is short for MacConkey. And as you can see in the picture, it is normally a light red colored agar plate. And this media is both selective and differential. So let's review what those terms mean. Selective means that only select organisms can grow on this media. And that's because the media contains ingredients that are going to kill or prevent other organisms from growing on it. In the case of a MAC plate, the ingredients that are the selective ingredients are crystal violet and bile salt. These two ingredients are going to kill gram-positive organisms. So we would say that MacConkey Agar selects 4 gram-negative organisms because they are able to grow on this plate, and MacConkey Agar selects against gram-negatives because they cannot grow on this plate. So whenever we're looking and thinking about our selective observations, what you're really looking for is growth or no growth. And because we're talking about an agar plate, we're going to be looking for the presence or absence of colonies. Now, this media is also differential, which means of the gram-negative organisms that can grow on the MacConkey agar, we're able to differentiate them based on their ability to ferment the sugar lactose. Some gram-negative organisms can ferment lactose, and some gram-negative organisms cannot ferment lactose. So we're going to be able to differentiate between those two groups of organisms. And for your unknown flowchart that you're working on, this is going to be the initial media that you do to divide your unknown organisms into 2 groups, because in this case, our 2 groups are the lactose fermenters and the lactose non-fermenters, okay? So how can you tell whether or not an organism can ferment lactose just by looking at the growth on the MacConkey agar? Well, remember, fermentation produces acidic byproducts. So there's going to be a decrease in pH if the organism can ferment the sugar lactose. So if we want to be able to visualize that pH change, we need a pH indicator. And in this media, we include the pH indicator neutral red. Under acidic conditions, neutral red turns hot pink. But when the pH is neutral, it's going to be the color that you see on this picture, that normal light red color. Okay, so let's take a look and see what would happen if we streaked different organisms on a MacConkey agar plate. So remember, this media is going to be selective for gram-negatives, and it selects against gram-positives. So if we streaked the gram-positive organism, Staphylococcus aureus, you would get no growth, right? Because Staphylococcus aureus is gram-positive, so it cannot handle the bile salts and crystal violet dye that's present in the media. So what do you see? No growth. That result would be negative, and your interpretation is that the organism cannot withstand the bile salts, the crystal violet, because it is a gram-positive organism. So this is one possible outcome if you streak something on a MacConkey agar plate. Now for your flowchart, you don't want to include this image and these observations, results, and interpretations, because your flowchart is only for unknown organisms. And in this unit, all the unknowns are gram-negative, so they can all grow. But this is good to remember for your lab quizzes and your lab exam. So let's take a look at the 2 possible outcomes for if you streak a gram-negative organism on a MacConkey plate. So looking at both of these pictures, you can observe growth, okay? You can see nice pink colonies on this left-hand picture, and you can see all the little dots here, those colonies, on the right-hand picture. So part of your observation for each of these are going to be growth or colonies, okay? Because you can physically see that. Sorry, y'all. So that's our selective observation, is growth. This organism or these organisms were able to grow on MacConkey because they're gram negative. Your other observation is color or lack thereof. On the left-hand side, you see bright pink colonies, but on the right-hand side, you see colorless colonies. Yes, that's your differential observation, is what color are the colonies. So looking at these two, which one is going to be a positive result and which is a negative result? Now, because they both are gram negative and they were both able to grow on this media, you don't need to give the selective result because they're the same. That doesn't help us differentiate them, right? We need something that's different between them. And in this case, it's going to be the color of the colonies. The color of the colonies is going to allow us to determine which group the lactose fermenters or lactose non-fermenters a given organism belongs to. So we only need to report that differential result because that's going to be what helps us differentiate these two organisms. So the hot pink colonies, that observation is going to be a positive result because this indicates that the organism is able to ferment lactose. On the right-hand side, it's a negative result because that organism cannot ferment lactose. So whenever you're trying to give the result and the interpretation, always think back to what we're trying to learn from the media. When we streaked, for instance, our unknown organism on this plate, we already knew that they were gram negative, okay? But we want to know, can it ferment lactose? So I always keep that question in mind because you can think of a positive result as the answer yes to your question and a negative result as the answer no to your question. So if something can ferment lactose, we know the pH is decreasing and that pH indicator is going to turn hot pink. So that's why the hot pink colonies indicate a positive result because it means yes, the organism can ferment lactose. Whereas on the right-hand side, those colorless colonies are a negative result because it tells us the organism cannot ferment lactose. Okay, so again, for result, you're only putting positive or negative. The interpretation is where you're saying what it means. Okay, but your interpretation does always want to be comprehensive and address the selective and differential observations that you originally made. So I'm going to walk you through it again for the left-hand picture, and you're going to apply this knowledge to filling out the right-hand picture, okay? So again, for the left-hand side, your observation is going to be hot pink colonies. The colonies indicates growth, and so that's the selective part. The hot pink is your differential observation, okay? So hot pink colonies, that's your observations. Your result is simply positive, and your interpretation is going to be the organism is a gram-negative lactose fermenter. Because again, gram-negative was the selective part. Only gram-negatives can grow. And then the lactose fermentation addresses your hot pink observation. Okay? Now, please notice on the slide, I said an example is E. coli. E. coli is not the only organism that will grow like this. It's just an example. Okay, there are actually several gram-negative lactose fermenters that grow this way. This is just an example. And on the right-hand side, there's lots of organisms that will produce colorless colonies. Salmonella enterica is just one of those. So with this media, you always want to make sure that you streak for isolation. You're going to do that streak plate technique. And that's because some organisms are very picky when it comes to producing that hot pink color. One of those examples is going to be Klebsiella pneumoniae. As you can see in the picture, the colonies do turn pink. Now you might say, well, I don't know if that's hot pink or deep pink. Don't overanalyze it. Okay, for this media, if the colonies are pink in any capacity, it's positive. Okay, that means it's a lactose fermenter. But look at this plate. you can see how the isolated colonies have that clear pink color. But in the areas of the plate where the colonies are all just mushed together, you do not see that pink color, okay? Because some organisms, just when they're all crowded and stressed out, you're just not going to be able to see it. And so in lab, one of the biggest problems I see people make, or biggest problem that they encounter, is when they initially streak their unknown on the MacConkey plate, they don't get isolated colonies. There's too much growth in all of the quadrants. And so you can't tell whether or not the organism can ferment lactose because you don't have any individual colonies to assess. So getting isolated colonies is key for success when streaking something on a MacConkey plate. All right, so real quick, thinking in terms of the unknown assignment that we have, I mentioned in the very first like big picture video that we need to divide our organisms into two groups, okay, the lactose fermenters and the lactose non-fermenters. Of our 8 unknowns, we have 3 that are lactose fermenters, that's the LF group, and then 5 organisms that are lactose non-fermenters, that's our LNF, if you're looking at this slide. Now, to help you remember which 3 organisms belong in the lactose fermenters, we call them EEK, E-E-K, because E. coli, E. coliaceae, K. pneumoniae. So EEK, kind of an easy way to remember that. All the remaining organisms that are not lactose fermenters, they all start with either a P or an S, so we call them the P cubed S squared group. All right, y'all, that is the end of this video. Let me know if you have any questions.